to SNEV Characterization. Welcome to SNEV Characterization series, and thank you for joining. My name is Maimouna Al-Musawa. I'm a PhD student at CTERM UKM Malaysia. Today I'll be moderating the talk along with my friend Eunice Bear, and we have a special guest, Dr. Claudio. He's going to give us a talk on how to navigate the storm, navigate the storm, a guide to EV analysis. So let me introduce Dr. Claudio. Dr. Claudio recently received his PhD from Ghent University. He's currently a postdoctoral fellow at Laboratory of Experimental Cancer Research, Ghent University, Belgium. He's been an active member of Train EV program. He was part of Train EV Conference Organizing Committee in 2021. He was also elected representative of ECRs from Train EV Consortium from March 2018 to 2019. He was also a trainer in EMBO course that took place in Heidelberg, Germany last year, and also co-moderated EMBO Technology Day on Technologies for Extracellular Vesicle Research in November 2020. He received a number of grants and awards, and also he has a list of interesting publications. He was an invited speaker to ICEV EV Club in October 2022, where he talked about the identification of EV reference genes for RT-qPCR. So Dr. Claudio, thanks for accepting our invitation. We're very excited for the talk. So you may start your presentation.
All right. Thank you, Maimouna, for the invitation and for the nice presentation. So hello, everyone. My name is Claudio Pinheiro. I'm a postdoctoral researcher here at the Laboratory of Experimental Cancer Research in Ghent University, Belgium. And like Maimouna explained, I recently finished my PhD fellowship, where I focused myself in understanding and establishing tools for increasing the rigor and standardization of the extracellular vesicle field. And in order to do so, what I did was I enhanced my knowledge on how to actually separate extracellular vesicles, how to understand what are the idiosyncrasies of the way that we separate and we prepare extracellular vesicles, and as well as we characterize extracellular vesicles as well, which led to several publications and several important informations for that. And so the name of my talk today is Navigating the Storm. And the reason why I called it Navigating the Storm is because, like most of you, I also started my PhD very unprepared when it came to extracellular vesicles. So actually, this is when I looked at some pictures on Google, and I Googled Navigating the Storm, I got this picture, which I thought was very appropriate to how I felt dealing with my research project in the beginning of my PhD. So actually, this was me here, and I was, as you can see, very unprepared. I was essentially in this box, and I could see my PhD project coming, but I didn't really knew how do I even start by analyzing extracellular vesicles. Where do I even start, and how do I go from here on forward? But luckily, there are several courses and several tools that you can use to your disposal. And I would like to preface my presentation by presenting you these tools. So these are the MOOC, so the massive online open courses from Coursera. There you have the MOOC 1 and the MOOC 2, which the first one is on basic extracellular vesicle analysis, from extracellular vesicle preparation to characterization. MOOC 2 is more focused on specifically health and diseases, and also how the vesicles play a very important role in both health and disease status. And last but not least, we have the MOOC 3, which is freely available on YouTube for everyone to follow. And this one specifically talks a lot about the detection and isolation of extracellular vesicles. And a lot of the information that I'm going to give you today, you can find more in-depth information about the preparation methods that I will talk about in these Coursera courses and these MOOC courses. But if the online thing is, if online courses are also not your thing and you would like something that is more hands-on, luckily we also have established two at least practical courses. We have the EMBO practical course if you're from Europe and you'd like to follow more hands-on courses. So these were performed last year, and that's where I met also Maimona, where we actually teach you how to extract extracellular vesicles from matrices and also how to characterize them. So this is a more dedicated hands-on course to really newcomers and also more experienced people in the field. And we really teach you how to perform that. And the same way as the workshop that you have most recently on also how to separate and characterize extracellular vesicles, but this time this one is in the United States. And I really incentivize those that would like to know more about how to prepare and characterize extracellular vesicles to apply to these courses, because this panel of starstruck people, these are all experts in the field, and it's one of the most renowned experts that you have contact with. And it's a very, very nice way and very nice environment for you to learn how to deal with extracellular vesicles. But for the topic of today's presentation, I will go over everything that I learned during my PhD, which actually resulted in the creation of this Nature Review Methods primer, where me, together with my colleague Lynn and my PI An, together with all other co-authors, we created this comprehensive review on how to prepare, characterize, and also apply extracellular vesicles, alongside all the tools that are actually currently available for you to actually prepare vesicles, so remove them from the biofluid source that you want. Also, the tools that you have available to characterize them and perform the proper quality control according to also MySIP guidelines. And then when you go from here and how to apply them in your day-to-day basis. So the topic of today's presentation will be mostly revolving around this publication, but for specifically today, I will focus more on how to prepare extracellular vesicles. So what are the methodologies that you have available to prepare extracellular vesicles, and how you can go from here, and what are the specific strong points, so that what are the pros and cons of every methodology that you have, and how can you use it to the best possible way to your specific study goal. So if we're all here in this specific point, we all know that extracellular vesicles have a very wide range of applications. Either it being at the bench level or at the application level, extracellular vesicles are currently being studied worldwide for their diverse applications. So at the bench level, we have researchers studying extracellular vesicles for their structure and composition, where people are looking for proteins, nucleic acids, lipids, their size, the density, their number. And we also have their uptake, the release, and the function, really to study the biology and how these extracellular vesicle, or the release of extracellular vesicle, impacts the biology of the cells and the cells that are releasing these vesicles. But of course, we have a whole other range of applications that we can have when it comes to the diagnostics, where we can actually perform a biomarker analysis or a multi-analyte biomarker analysis using extracellular vesicles, since they completely represent the cell that has released them. So they're a fingerprint of the cell of the parental cell that released the extracellular vesicle. But we also have a whole range of potential therapeutic applications where researchers are trying to actually load cargo inside of extracellular vesicles due to their properties, due to their membrane and their capacity to sometimes avoid the immune system and also shield the cargo that they have inside of them. So throughout my PhD, I encountered several researchers worldwide that have a very specific way of going about their research question. So when you come to the first stages of when you want to study extracellular vesicles, the first point that you want to do most of the time is you identify an idea, and you have the idea that you would like to study extracellular vesicles in a specific biofluid, and you just go to literature, you perform a literature search, and here you identify what are the methods that are used to isolate the vesicles that are people that are studying, for example, RNA, and I would like to study RNA, what are people doing? And then I just simply take their blueprint, take their protocol, and apply it to the biofluid that I want to do. The thing is, this doesn't always work. And the problem, and I will explain why this doesn't always work when we talk about the idiosyncrasies of specific preparation protocols. However, what we would like to propose, and what we propose in our review, is that we first need to identify, of course, always your question, but then you select and explore the composition source to specifically design and implement a preparation protocol that tackles the components that are present in your source composition. After that, you should be able to completely remove or study EVs from other materials, so separate EVs from other materials, or also, if you so choose, so perform a bulk EV analysis or specific EV subpopulations where you're able to aliquot, store, and use your EV preparations at will. As such, in order to minimize what I just said, essentially what we propose is the following. Instead of having an idea and proceeding immediately to a literature search about it, you first have your idea. You always start from an idea. That's always a spark in the beginning. And from here, you move into identifying where do you want to study this extracellular vesicle. So what is your biofluid of choice? What is your source material? Once you identify your source material, you move into the literature search where you understand all the components that are present in your source material. And from this source material, you understand everything that comes out of it, and you can have a literature search of what is currently performed in your field of choice and in your biofluid of choice. Then from here, you can remove this blueprint, apply this blueprint, and then proceed for your experimental scenarios. So the best way that I have to explain this is by performing a very simple analogy when it comes to the EV toolbox that we have available. And when it comes to this toolbox that we have available, I like to always give an analogy to the toolbox that we always had at home. So at home, everyone has a toolbox where they have several different types of tools that they serve different purposes. So if you would like to, for example, hang a frame on a wall, we would use a nail and a hammer. And if you would like to paint a wall, we'll probably use a can of paint and a brush. But however, you wouldn't do the other way around. So you definitely wouldn't use to paint a hammer the same way as you wouldn't use a brush to nail something on the wall. And the same thing happens for extracellular vesicles. So there's always a toolbox as well associated to extracellular vesicle analysis, where you have extracellular vesicles, but you also have different tools that serve different purposes for the analysis of your EVs and your sources. And that's really important because actually extracellular vesicles are something extremely complex to study. So EVs, as most of you know, can be released by almost the majority of cells known to man, and these can be either from different, they can be from eukaryotic cells or also prokaryotic cells. So we have protistas, fungi, plants, EVs, mammalian EVs, bacterial EVs, and also archaea EVs. But this is not where it stops. Actually, there's so many more origins of heterogeneity, since it's known that extracellular vesicles can be completely dependent on the cell state of the cell. Being the cell healthy, diseased, or even engineering extracellular vesicles. Vesicles have different biogenesis sources or biogenesis routes, where you have then the forever known exosomes, ectosomes, apoptotic bodies. But then for the bacterial side of things, or the prokaryotic side of things, we have the OMVs or the EOMVs and the CMVs. And this is another level of complexity that we have, on top of the fact that we have source-specific extracellular vesicles, so they can come from the body ecosystem, but they can also come from the environment ecosystem. So here in the body ecosystem, we have the blood plasmas, the urines, the tears, and on the environmental ecosystem, we have the soil, the dust, the ocean water, or just water overall. And on top of everything else, we also have methodological heterogeneity. So how actually extracellular vesicle preparation methods actually impact the final preparation that you have, and how this actually in the end changes the way that you look at your extracellular vesicles. On top of all of this, vesicles can also be different in terms of structure. So they have different sizes, different densities, different surface charge, different refractive indexes, but they also have a different composition. So they can have different proteins, lipids, nucleic acids, metabolites, glycan, etc. And here in this table, you can definitely see how big this heterogeneity is at both the eukaryotic level, but also the prokaryotic level. So this really puts it in perspective how important it is actually to have a versatile tool set to cope with this level of diversity. So we would not be possible if you have only one tool to deal with all of this, because currently it is impossible that we have a one-fits-all scenario. We have to have a diverse tool set that actually is able to be expanded in order to deal with this EV heterogeneity. So in order to exemplify how this would work, I would start by giving you an example of one of the most complex biofluids from the body fluids that we have, which is blood plasma. So in order to retrieve blood plasma from a sample of blood, we first have to separate the blood plasma from red blood cells, white blood cells, and also platelets. Here, there's already a specific degree of importance because red blood cells and white blood cells and even platelets can release extracellular vesicles, which will end up in your blood plasma. But let's say that we're able to successfully remove all of these components from your blood plasma component. In your blood plasma, you'll still be able to find things such as cell-free nucleic acids, lipoprotein particles, abundant proteins, also aggregates, soluble proteins, and of course, your extracellular vesicles. And when it comes to the study of blood plasma, one of the most in blood plasma, the key components that you have to look for are specifically, obviously, your extracellular vesicles, but also the lipoprotein particles. And the reason for that is because most of the separation methods the preparation methods that we have currently try to prepare vesicles based on their characteristics, their intrinsic characteristics. And here in this publication, where researchers actually plotted the density and the size of extracellular vesicles alongside lipoprotein particles, so we have high density lipoprotein particles, low density, very low density in chylomicrons, as well as protein aggregates, we see that actually extracellular vesicles overlap with lipoprotein particles in terms of size, specifically LDL, VLDL, chylomicrons, as well as protein aggregates, but they also overlap in terms of density. So extracellular vesicles have equal densities as high density lipoprotein particles. To make things worse, the latest reports have reported something around, in circulation currently, for one vesicle that you will have around 1 million lipoprotein particles, which means that in order for you to accurately prepare an extracellular vesicle from blood plasma, you have around 1 million lipoprotein particles for one extracellular vesicle that you have to remove from your biofluid. So your preparation methodology needs to be very intrinsically prepared to tackle these problems that you have in your biofluid. And this, of course, is just blood plasma. If we put everything in perspective into all the biofluids that we currently have, or most of the biofluids that we currently have, you can actually separate things based on general components, source-specific components, environmental ecosystem, and also conditional components. And here, again, we also plotted the density and the size measurements of extracellular vesicles alongside all of the other components. And it's very clear that extracellular vesicles overlap with other components in terms of size and density. And here, it's really where your preparation methods actually come into play. So here is where you open your toolbox and you extract the tools that you want to tackle specific things that you want to remove from your preparation, from your source, in order to, in the end, have only, in theory, the extracellular vesicles that you intend to study. So today, I'm going to talk a bit about these preparation methods. And when it comes to the preparation methods, there's two different groups that very simplified we can discuss about. We can talk about methods in terms of concentration and enrichment. So concentration methods are essentially methods that aim to reduce the volume source while retaining extracellular vesicles from other materials. So these are very handy methods when you start from a very big amount of source material and you want to decrease this volume in order to enhance or enrich the application of extracellular vesicles. And then we have enrichment methods. And enrichment methods, the whole aim of enrichment methods is to retain extracellular vesicles or a particular extracellular vesicle population based on size, density, whatever you choose to study, while reducing the amount of other materials. So I will go over every single one of these methodologies, but before I do so, let's talk about performance metrics. And the two most important performance metrics that we have in the field, and for both newcomers and also advanced researchers in the field overall, are efficiency and specificity. And when it comes to the study of efficiency and specificity, it's always a balance. You always tend to gain something more or something less. If you gain on efficiency, you tend to lose on specificity and vice versa. So what is efficiency and what is specificity of preparation methods? Well, efficiency is the extent of which a method can recover extracellular vesicles from the present source. So this is often called recovery. So this is the capacity that your methodology has to recover the maximum amount or recover an X amount of vesicles that you have from your original source. And then on the other side of the coin, you have specificity. Specificity is the extent of which the method can selectively separate and eliminate other components from your extracellular vesicle preparation in the end. And this is often called purity. And the way that you can see this is that if you have an extracellular vesicle preparation, most of the time, these are inversely correlated. These two performance metrics are inversely correlated. This means that in most of the time, in a scenario that you have a high efficiency, meaning that you have a lot of extracellular vesicles in your preparation, this usually means that you have a low specificity of your preparation, here depicted by the presence of other components other than your extracellular vesicles. However, if you move towards a higher specificity, so if you increase the purity of your sample preparation, it tends to decrease the efficiency, meaning the recovery of extracellular vesicles that you have in the end. Ergo, you have less vesicles in the end, but as a consequence of that, you also will have less contaminants in the end, allowing you to study better the extracellular vesicles that you intend to do. So now I'll go over every single one of the methodologies and provide you with pros and cons of each methodology so that then we can discuss it further down. So first, let's start with concentration methods based on size. These are your filtration methods. The filtration methods that you have, so again, concentration methods, the whole overall identity of a concentration method is to reduce the volume that you have. So the initial input volume that you have and reduce it to a minimum amount so that you can start from that and proceed for downstream analysis or even further preparation from your sample. So usually these filtration methods are based on either dead-end filtration, where your sample goes through in a vertical way through the filter, or tangential flow filtration or cross-filtration, where your sample moves parallel to the membrane that is placed horizontally from the sample, and the sample is then filtered based on that. So I will always present here as well the size ranges of extracellular vesicles and potentially also other components. So most of these preparation methodologies in terms of concentration are very commercially available, and that's one of its cons. One of its pros, I'm sorry. So the pros of these methodologies that are commercially available, so it's very easy to have access to these methodologies, and they're very easy to implement and use. So they don't have a very high levels of entry. You can simply acquire these. Most of these commercial methods are very easy to apply because you can simply acquire these methodologies and then apply them in your day-to-day basis. They have a very low hands-on time, most of these, so you just have to either sometimes you use centrifugal forces, so you can just put them on a centrifuge and run the sample, and they're very high efficient methods, meaning that they recover a very, very high amount of vesicles in the end, albeit, of course, with a low specificity as well, and that's one of its cons. So because it's a concentration method, it's not very specific to the separation of extracellular vesicles, meaning that you enrich a lot of vesicles, so you have a lot of vesicles in the end, but you also have a lot of other components that you have in the presence of it because it's not able to separate things based on the vesicle characteristic itself, just the size, and that is usually a problem. So because of that alone, these methodologies don't usually unlock the pure study of extracellular vesicles, because if you really want to study an EV-associated application, this is usually not the way to do it because you have other components that can actually affect the way that you perceive your results. So you can actually think that you're seeing a result based or an effect based on the vesicles, but actually is the other components that are providing its effect. Lastly, dead-end filtration based on the biofluid that you use can also be susceptible to clotting. So this can be a problem where you have to replace the filters multiple times, and ergo you will lose a lot of sample as well because the filter is clotting and you're losing sample because of that as well. So this is concentration based on size. Let's talk about concentration based on volume exclusion. And the volume exclusion is one of the most renowned known concentration methods that exists, which is it uses a hydrophilic polymer to tie the water molecules of your sample and finally precipitates everything down in your sample. So these are also commercially available, but you can also implement them in the lab by the use of polyethylene glycol. So there are several publications that do so. And it's very easy to use and implement as well, and it has a very low hands-on time. So most of the time it requires an overnight step, but then you're done. It's one of the, or if not the highest efficiency method that you have. So most of the time you will recover actually almost 100% of the vesicles that you have in your biofluid source. However, as a consequence of that as well, we have a low specificity associated with this methodology. And again, alone, it will not really unlock the study of extracellular vesicles. And once again, depending on the biofluid, it can generate insoluble pellets. So meaning that because of the way that the polymer works, if the biofluid or the source material contains a lot of other things that can affect the pellet composition, sometimes it can be very hard to dislodge this pellet and resuspend it in lower volumes. So this is definitely a con. So when it comes to our grand scheme of things, where would we put concentration methods? So usually speaking, concentration methods are at the high efficiency, low specificity spectrum of things. So these would be, again, your concentration methods for both size and volume exclusion. Now let's talk about enrichment methods, specifically the size enrichment methods, which are your DUC, your differential ultracentrifugation, sometimes also called as sequential ultracentrifugation, your size exclusion chromatographies, and your asymmetric flow field flow fractionation, or AF4 for short. These methods tend to enrich extracellular vesicles based on their size and tend to sometimes even fractionate the sample based on their size. So that you'll see that this is one of the pros of these methodologies. So I think it requires almost no introduction, but differential ultracentrifugation is by far one of the most known preparation methods that most people are aware of. And it uses centrifugation steps, so successive centrifugation steps at increasing G-force and or time to enrich extracellular vesicles based on their size and on their mass. So really uses and exploits the precipitation, the size and the size of the vesicles and the mass of the vesicles to precipitate them down faster than the other soluble proteins or other proteins that you have on your sample as well. The thing is with differential ultracentrifugation is that it's very nuanced to the speed, the rotor, and the time that you use to spin most of these things. So in most of the scenarios, if I were to use differential ultracentrifugation with a 5,000 G, 10,000 G, 20, and 100,000, I have a specific set of results. But it can happen that someone else uses just 10,000, 30,000, and then 80,000 or 200,000, and the results are different. And because of that, there's several different types of protocol regarding differential ultracentrifugation, and there's no specifically a consensus in which are the typical steps of a differential ultracentrifugation, with, I guess, the exception of the last step, which is the 100,000 Gs. Next, we have size exclusion chromatography, and size exclusion chromatography, I think it's also becoming one of the most known type of enrichment methods that we have in the field. So the way that size exclusion chromatography works is usually based on a column that is filled with a porous gel filtration polymer. So these are usually beads, porous beads. And the way that it works is you load your sample on top of these, and your extracellular vesicles, if they're bigger than most of the components, they are able to cross through the beads in a very linear way. However, because the beads have pores, components that are able to penetrate the beads will have to go through a different path, and it's a much longer path. So these components will elute later, while your vesicles of specific sizes will also elute sooner. The thing with size exclusion chromatography is, once again, you have different nuances because you can also have different. So if in this case the polymers or the porous gel is different sized, you can also use 70 nanometer sized, you can use 40 nanometers. So you can really filtrate and fine tune your extraction based on the size of the pores in order to achieve the best separation that you possibly can, or specifically according to your study goal. AF4 is one of the most newest or one of the most recent techniques that we have, and it uses two perpendicular flows with a channel, with a membrane, so that you can separate your vesicles. So similar to size exclusion chromatography and differential ultracentrifugation, it uses size, but this time it uses a perpendicular flow. So it has a flow vertically, which pushes the sample down, and then it has a flow horizontal, which pushes the sample alongside the membrane. And this allows for the separation of the extracellular vesicles from other components with different sizes as well, allowing you for really a fractionation of your sample based on the size of the vesicles. In terms of pros and cons of these methodologies, most of these methodologies are extremely efficient at removing sample or components that do not share the same size as vesicles. So examples of these are soluble proteins and protein complexes, or for example, in the case of blood plasma, HDL from the rest of the vesicles, because HDL has a smaller size in terms of smaller size compared to extracellular vesicles. On top of that, using these methodologies actually unlocks the analysis of vesicles based on their size. So this is where you unlock really the use of nomenclature such as small vesicles, medium vesicles, and large vesicles. And you will notice that most of the time, the publications that apply these methodologies, or that actually have these nomenclature, actually apply these methodologies for their study, specifically. Alongside the use of the AF4, for example, allowed for the study of, or the discovery of actually a new nanoparticle, so that is the exomeres, and this is also only solely based on this technology and this new generation of technologies. In terms of cons, of course, everything has a con. These methodologies are able to, again, separate particles that share the same size. So, for example, very low density lipoprotein particles share the same size as extracellular vesicles, and ergo, because of that, these methodologies are less efficient when it comes, or less specific to the removal of the vesicles compared to other components that can be there. And as well, specific size methodologies can affect the EV integrity. So they can actually affect the extrafacial surface. So this is really more in the sense of the protein corona and the components and the bimolecular components that are on the surface of the extracellular vesicles, as well as some methods that are also known to create some aggregation of your extracellular vesicles in the end. Next, we have the density exploitation methods, and these are the density gradients and the density cushions. So density gradients exploits the intrinsic buoyancy or intrinsic density of extracellular vesicles, and how this differs from other components that can be present in your source material. So density gradient usually enriches vesicles by laying a sample either on the top or on the bottom of your density gradient. And upon centrifugation, your particles or your vesicles will migrate to their density, to their intrinsic density, while other components will migrate to their intrinsic densities. And because of that, you will be able to accurately separate them properly or efficiently separate them from other components. On the other side, we also have the density cushion, which is another version of a density exploitation methodology, which enriches vesicles by using a layer of density medium, which is usually smaller, but overlaid with a large sample volume. And this allows for a higher input compared to, for example, density gradients, where the sample input is very, very low. Pros and cons of these methodologies is that, once again, these methodologies are extremely efficient at removing other components that do not share the same density as extracellular vesicles. So these are your protein complexes or your cell-free nucleic acids, low-density lipoproteins, etc. And they're often used because of their specificity, because of the achieved specificity that they have. They're often used as a control for EV preparations. So there's several instances where a density gradient can be used as a control for your sample to really see if the effect that you're seeing from your extracellular vesicles is due to your methodology, or can be also perceived by the use of density gradients. However, they also are cons. So in terms of cons, most of these technologies require a moderate to advanced technical prowess in order to perform. So there's a very high level of entry in order to perform these methodologies. It's not that easy to perform, and you require some premeditated knowledge in order to perform. And on top of that, because of this high advanced technical demand, most of the time these methodologies are also very operator dependent, meaning that they can change a lot depending on who is actually performing the technology. Also, the removal of the density medium in which the sample is mixed with requires another application. So most of the time you actually need to remove this density medium, because if the density medium is present in your sample in the end, it can actually affect your downstream analysis and the way that your analysis is going to be read out in your characterization methodology. Now, last but not least, we have the surface exploitation enrichment methodologies, where they use the negatively charged or the antigen present at the surface of extracellular vesicles. So when it comes to the charge of extracellular vesicles, as most of you surely know, extracellular vesicles have a slightly negative zeta potential, so they're slightly negative charged, and that allows for the exploitation of this charge to separate them from positively charged material. And this can be in forms of chromatography. So, for example, anion to cation exchange. They can be bead-based, and they can also be membrane-based. So it can also be through a passage on membrane. But they also have antigen. So most of your extracellular vesicles have, as most of you know, the tetraspanons, so the CD9, the CD63, the CD81s, and actually you can actually capture extracellular vesicles based on this using an antigen-specific antibody captured to a bead. And that really allows for this affinity capture, which enriches in specific EV populations, of course, as well. Because most of the time, what happens here is that you load for a specific antigen, so let's say CD63, and then subpopulations that are CD63 negative, meaning that they don't express CD63, so they don't have CD63 present at the surface of your vesicle, they will not be present in the end. Pros and cons of these technologies, we can also always achieve a very high level of specificity, especially when you use affinity capture, because in theory, they will only be present, for example, if you use CD63 again as an example, only particles with CD63 positive at their surface or at the membrane would be captured. And this has a very high applicability in terms of biomarker detection or any diagnostic potential. So these are actually one of the most foreseen methodologies when it comes to the diagnostics or the use of diagnostics for extracellular vesicles, because of their capacity to retrieve vesicles based on this antibody, based on the use of antibodies. In terms of cons, the elution, this is always a problem as well with these methodologies, because the elution of the EVs capture in these methodologies is not that straightforward. And if you want to capture the vesicles based on this, but then you want to release the vesicles for a downstream application, this is usually not that easy, and most of the time involves the release of the antibody itself. So for example, if your overall idea is to perform, let's say, proteomics or mass spec, and you want to analyze or you want to retrieve the vesicles, if this also retrieves the antibody, you're going to have a very high degree of, let's call it, contamination from your antibodies, because they're also going to be present in the sample in the end. Of course, one of the definitely cons of these methodologies is that sacrifices EV populations due to the very restrictive enrichment procedure, meaning that if extracellular vesicles have a slightly positive charge compared to the charge that we're actually collecting, we will miss those vesicle populations, as well as antigen present or not. We also saw it as an example that I gave with CD63. So once again, in the overall scheme of things, the way that you can perceive this is that actually enrichment methods are a bit more towards the higher specificity, but of course, because of that, they sacrifice a lot of the subpopulations due to their strong points to remove extracellular vesicles or move extracellular vesicles from other components based on either their size, their density, their biophysical characteristics, or biochemical characteristics. So actually, enrichment methodologies are a bit in the middle of our schematic of the overall grasp of things. So by now you're probably thinking, okay, but what if I really want to study just vesicles and I don't want to study anything else? So how can I get something that is very highly specific, but I don't mind having not that many. So you'll have not a lot of vesicles, but you'll have a very high degree of specificity. So what are the methods that do this? And these are actually your separation protocols. So separation protocol is actually characterized, or is actually, you can see it as a sequential order or a sequential application of enrichment methods or concentration methods. So this is the application of different methodologies back to back in order to achieve a higher separation of your or a higher enrichment, a high specificity of your extracellular vesicles. And for that, I created this schematic over here. So here what you have is potential combinations for example, blood plasma, using blood plasma as an example. You have potential combinations using, for example, a volume excluding polymer, so concentration, so this is precipitation based, differential ultracentrifugation, which exploits the sedimentation based of extracellular vesicles, iodexanol based, which is based on their density, so this is the use of density gradients, and then column based, which is the use of size exclusion chromatographies or binding elution chromatographies. And I can imagine this is a quite complex figure, but if the way that this works is that you can see what are the components that are retrieved based on specific methodologies and the use of methodologies. And if you are to apply them in a very specific order, what you can actually do is achieve a preparation of extracellular vesicles with a high degree of specificity, albeit that you're sacrificing the efficiency because of these methodologies. You're putting methods back to each other, and you're losing a lot of vesicles in the end because of that. But in the end, the vesicles that you have are very pure, and they are devoid of any other components. So as an example that I like to give, if you were to apply, for example, size exclusion chromatography to blood plasma, you will essentially capture most of the things, but you will lose the HDL, so the high density lipoproteins, because they don't share the same size as vesicles. And if afterwards you are to apply a density gradient, you can actually capture medium vesicles, or let's say medium-sized vesicles and small-sized vesicles, and you will not capture HDL because they're not there in the beginning. And that's really the example that I would like to give, because that's really what we did also, and that's also what we do here in Ghent. And we actually created a protocol and where we start from blood plasma and we apply blood plasma on top of a size exclusion chromatography, and we collect what we call the crude extract. And the crude extract is what elutes from the size extrusion chromatography, where we know that we're going to have extracellular vesicles, but we also will have other lipoprotein particles. So here depicted in black arrows for the lipoprotein particles and white arrows for the extracellular vesicles. But because we know that this is the case, what we then do is we take this crude extract and we apply it on top of a density gradient. And this really allows for the accurate separation of these both components that we still have. So because we here deplete the high density lipoprotein particles, which are the particles that overlap with the same density as extracellular vesicles, we then are able to separate extracellular vesicles with a very high specificity, so very high degree of purity, as you can see here highlighted by the white arrows. And then we're able to also accurately separate the lipoprotein particles depicted here in black arrows. Nevertheless, we're not the only ones to do this, and I'm not here to definitely depict that we're the only ones to do this. There's several publications that you will be able to find throughout literature that actually conduct these sort of experiments, that perform sequential implementation of preparation methods in order to achieve preparation of vesicles with a high degree of specificity. So in the grand scheme of things, actually, separation protocols or the separation methodologies actually are the ones that allow you for the very high specificity with the lowest efficiency. However, but the vesicles that you have in the end have a very high degree of purity, meaning that you're able to study them very, very well. So, and just to give you a small little example how impactful it is, that if you choose the wrong preparation method, this can affect the way that you see your preparations and actually your study of vesicles. In 2014, our lab actually performed a small pilot study where we actually analyzed 4 preparation methods, and we removed the vesicles, so we isolated vesicles using these 4 methods, and then we studied their RNA profile. And what we could see is that the RNA profile was extremely different in most of the methodologies, and except, for example, here in density gradient, which was more or less the same. All the other ones actually intertwined in terms of each other. And actually, what we then saw when we performed an analysis was that most of these methodologies that that the profile is more similar than the density gradient, they contain Argonaut-2. And Argonaut-2 is known to contain RNA strands inside of it, and this can actually affect the way, clearly the way that we're perceiving the RNA profile of our samples, and ergo affect really the way that we're seeing this, that we're seeing their vesicles. So this is just an example with 4 methodologies. But actually, in 2017, when we created the EV-Track knowledge base, what was done was that we analyzed 1,226 EV publications, and we reached the conclusion that out of these 1,026, we had almost 1,038 unique protocols, meaning that you can actually expand this that we found with 4 methodologies with almost 1,000 unique protocols. And if we are not careful, we can lose ourselves trying to study extracellular vesicles, but we'll never be able to do so because we keep reinventing the wheel trying to understand how this test works. So overall, as a take-home messages, I would like to highlight some key parameters. First of all, in terms of performance metrics, here I only specifically talk about specificity and efficiency, but there's so much more that you can look into. And overall, as I started this presentation, there's no one key fits all scenario. All methodologies have strengths and weaknesses, and in order to avoid these weaknesses, the best thing that you can do is understand your biofluid and select your method based on your specific study goal. And as such, I would like to welcome you to remember these following steps next time you're trying to establish a study, a project, or even go about your experiments. So first, identify your study goal. Understand why you want to study the extracellular vesicles and what you want to study about the extracellular vesicles about. This will allow you to specifically determine if you need a high degree of specificity or efficiency or something else. 2, investigate the biofluid composition. So definitely understand in your source what is present alongside the vesicles and what can you do to remove these things so that you only in the end have only extracellular vesicles or something else if that is your goal, of course. Number 3 is to select the appropriate method which allows you for you to achieve that goal. So that's where it comes, this knowledge that, for example, today we talked about regarding the pros and the cons of each methodology. So here's where you really enter regarding the knowledge that you have based on literature about the strengths and weaknesses and also the performance metrics that you have alongside with these preparation methods. And then, of course, proceed with quality controls and downstream applications of your EV preparations. So overall, I also want to highlight that I only specifically talk about EV preparation, but this is a one step on a multi-level puzzle. So this is really 4D chess that we're playing here, because then comes the characterization and everything that comes with characterization regarding that before your even downstream analysis. And I will not speak too much about this, but I would like to highlight when it comes to characterization, we have also a lot of other tools available to us. And these can range from different output information to assay duration, as well as different parameters that you have or different performance metrics that you have regarding these methodologies as well. And if you want to study them based on single EV analysis, bulk EV analysis, if you want to measure concentration, if you want to measure biomarkers, this is things that you can definitely use. And this is another level of complexity that we have on top of just the preparation methods that can give a lot of heterogeneity. So to wrap up this presentation, I would just like to highlight that once again, I think this is a very exciting field, and most of you should be very excited for the study of extracellular vesicles, whether it being a bench application, or at the application itself. So the study of the biology of extracellular vesicles, or potentially applying them to real biomedical purposes. And for that, I would like you to always remember that alongside it, you have a specific tool set that you have with several tools that you can use to the best of your knowledge. And if you use this to the best of your information, to the best of your knowledge, I hope that in the end, you will not find yourself in a position how I find myself in the beginning of my PhD, and that in the end, you will be able to have a smooth sailing and be able to navigate this storm, which we call the extracellular vesicle analysis. Lastly, I would like to thank you all for your attention, and also highlight that we are currently looking for PhD candidates and also postdoctoral fellows. So if anything that I've discussed here today, or if you'd like to learn more about this, feel free to send on an email, and we'll go about from there. Thank you for your attention.
Okay, thank you very much, Claudio, for the talk. Very, very appreciated. As mentioned by Mamona, the talk has been recorded and will be present on the SNES YouTube page. Also, for those in the audience, please do sign up to be a member of the SNES community. It's free, and you get a lot more information and more future talks and seminars also. Okay, so I'm going to start with some of the questions present in the chat. So the first one's by Agnieszka Razin. She asks, how do you get rid of the hydrophilic polymer from your sample? I'm guessing this refers to after PEG isolation.
That's not always easy, but you can definitely do as well. Most of the time, the protocol requires a washing step, and this washing step usually is enough. But what you can also do is you can dilute your sample, and pellet it afterwards. This usually allows to the removal of the excluding polymer, but this usually is done by resuspension of the polymer. If you resuspend the polymer itself, it will dissolve, and you can treat your vesicles afterwards. But this is again a concentration method. Usually what it involves is that ideally you use a concentration method, then followed by an enrichment method. So the concentration method, the sole purpose is really to deplete the volume, and then if you resuspend it in a smaller volume, you can apply a specific enrichment method based on that, and that should assist you also in the removal of your excluding polymer.
Okay, thank you. I apologize once again if I mispronounce anyone's name. So the next question is by Marianne. She asked, what's the difference between density gradient and differential centrifugation, in the sense that what's the best criteria to choose one over the other? And then following on from that, how do you get rid of the sucrose or glycerol?
Okay, yeah. So those are two different questions, right? In terms of which one, how do you go about choosing which one? That's really what is hard about these presentations about preparation methods, is that most people want to know which one is the best for me, and that's very, very study dependent, right? That's usually my first question is, what do you want? Why do you want to study extracellular vesicles? What do you actually about extracellular vesicles would you like to study? How important is it that in the end you have the highest pure vesicle preparation that you have, or is it okay that you have something else, right? Because if you're going to only perform a Western blot, that doesn't really matter, right? As long as you know that you want to detect for C63, etc., that's fine. If that's your idea, that's all okay. If you want to get something more done, that's different. Of course, for example, if you want to study different subtypes of vesicles based on their size, a density gradient would not suffice for this, right? A density gradient is exploiting the intrinsic buoyancy density of the vesicles, and this does not separate based on size. So for that, I would say definitely use something such as differential ultracentrifugation. But please be aware that there's downsides to this technique as well. Regarding the second part of the question, which is the removal of the sucrose, I think that was the question.
How do you get rid of the sucrose or glycerol? Yes.
Yeah, so in our case, what you usually do is we apply size excluding chromatography following the density gradient. That's what we do here in Ghent. But we can also do is you again dilute the sample. So if you have, for example, your fraction where you have your vesicles is 1 milliliter, you dilute this into around 15 to 16 millimeters, and then you precipitate everything down. And this usually dilutes the sucrose and allows you to, in the end, retrieve your sample with still some remnants of sucrose, but not as much that you will affect the downstream analysis of your sample.
Next question's by Pamela Valva. She asks, to obtain the EV from plasma, is it completely necessary to use plate inhibitors? And then also, can the EVs obtained from the plasma be preserved at minus 80 degrees Celsius?
Can you repeat the first part of the question, please?
So the first part is when you obtain EV from the plasma, is it completely necessary to use platelet inhibitors?
No. You meant PEG, right?
Maybe I'm assuming here it says platelet inhibitors. I'm sure she might mean.
No, that's definitely not the case. We don't do it ourselves, so that's not the case. Regarding the second part of the question, in terms of
in terms of storage, right?
Yeah, that's possible. We do it ourselves. That's also something that we discuss in the review, so which is the storage conditions, and that's also something that is very commonly discussed in the field. There's several ways that you have to go about and store your vesicle preparations, and this also changes the way that you then retrieve them in the end, or the way that you actually analyze them. We usually store them at minus 80 always, but if you want longer preservation times, there's things that you can do to increase the preservation of your vesicles, of course.
Next question is by Israeli Noy. What is the best method for in terms of downstream for proteomics?
Okay. So when it comes to the analysis of proteomics, that's the case is, yeah, I think you had Professor Suresh really talking about this as well, and that really goes about the way that what do you want to see in the end of your vesicle, of course, and the thing is with proteomics, I think one of the things that Professor Suresh mentioned at the time was if you have a lot of other things in your components other than just vesicles, so if your preparation, so if you have vesicles and, for example, in terms of lipoprotein particles, so many other lipoprotein particles, it is possible that the presence of these other components can overshadow specific components that you want to detect in your proteomics. So usually the best answer that I like to give in this, the best way to do proteomics, which I assume is to detect something EV specific, would be a methodology that has a very high degree of specificity, so that in the end, your claim, if you want to claim that this protein is related to vesicles, then you definitely know that the presence of this protein is due to the presence of the vesicles and not due to the presence of any other components or something that you caused during your preparation. So I would say something with a high degree of specificity.
And then just to correct myself, because I was wrong, so I apologize to Marianne. The DC she was referring to was not density, not to do with differential centrifugation. It was density cushion. So it's density cushion versus density gradient.
Right. So density cushion versus density gradient. Density gradient revolves around the use of a lower input volume, and this lower input volume then is placed onto. So essentially, the way that you can think about it, I think the best way that I have to describe it is you think about the ratio of sample to gradient that you have. In a density gradient, you usually have a very low amount of sample in a very large amount of density medium. In a density cushion, you have a very big amount of density of source material and a lower amount of density cushion. So the density cushion allows you to separate based on density, but usually not with such a high degree as a density gradient would do. However, a density cushion does allow you to put much more volume than a density gradient allows to. So it's really in the word of gradient versus cushioning. So cushioning is more like a soft pelleting, I guess I can say. Depends also how much time you centrifuge your sample, but the density gradient allows for a more accurate separation based on the density of your sample.
Okay, thank you. The next question is from Christian. He says, in the beginning of the talk, you said that exosomes perfectly represent the biochemical environment of the parental cell. However, isn't it true that some cytosolic particles are taken into exosomes by means of active mechanisms which consume ATP? And then this could generate a bias in the content of these EVs.
Right. So most of the time, and I'm in terms of the loading of proteins or actually any molecules inside of extracellular vesicles, I think this is quite simple in a sense that if it doesn't exist in a cell, it shouldn't exist in the vesicle, right? Because the vesicle is a non-self-replicating molecule, and it cannot create anything inside of the vesicle itself. So I think the way that you can go about this is that if it's present in your vesicle, it should be present in your parental cell. Of course, this then really comes into the biogenesis of the vesicle, because I think Christian also specifically remarked to the use of exosomes that this is definitely a different pathway than just the use of the term of extracellular vesicles that I'm having, right? Because exosomes really come from the endocytic pathway, and because of that, they can have different components inside of them or outside of them than something that comes directly from the plasma membrane. So these are things that you definitely need to pay attention to, but generalistically speaking, rule of thumb, if it's something that is not present in your cell, it shouldn't be present in your vesicle as well. Unless, of course, in cases of things like the bimolecular corona, if the extracellular vesicle is expelled to the extracellular milieu and then is in contact to a specific component, and that component then self-forms around the vesicle, that is a different story, of course.
The next question is by Delila Finelli. She asks, in terms of yield of EVs obtained from a freshly prepared sample versus a sample that's undergone freeze-thaw, and what is the amount or type EVs that kind of suffer the most from these freeze-thaw treatments?
Yeah, so it's a good question, because so the thing is, if you freeze thaw your sample several times, you're going to lose a lot of vesicles in the process as well. This is common to any biological component. Especially if you want to freeze cells, then the same thing will happen. So when you, for example, thaw a cell line, the same thing happens because you're subjecting the cell to a very stressful mechanic, and that really results in the bursting of cells or bursting of vesicles, and ergo you lose a lot of cells. If, of course, one of your highest points is increasing the yield, your best chances are always with fresh sample. So if you can always avoid a freeze thawing, perfect. That's why most of the people also, when it comes to the vesicle preparations afterwards, they aliquot their sample. So they try not to have their sample all in one go, because that way you avoid multiple freeze thawings. So if the idea is really to have the highest yield that you could possibly have, I would advise you to start with a fresh sample rather than something that is already frozen. But of course, sometimes that is unavoidable, and in that cases, avoid as much as possible freeze thawing.
I see a few people have asked, the same people have asked multiple questions, so I'm just going to skip a few and go to someone who hasn't asked the question before, so I apologize. This is from Santiago Moreno. He asks, what extraction method maximizes the small RNA content in terms of present in the EVs from the ones that you have tried?
Yeah, so small RNA content, there's several ones. The question is then, well, I think sometimes I'm going to become a bit of a broken record here, because I'm going to say a lot of the same things, but it really depends also on the but if it's small RNA, and then the problem is it's small RNA based on, I assume, from vesicles, then again, high specificity methodologies. So these are the methodologies that usually the separation methods that use combinatorial, so back-to-back, so combination of enrichment methods to achieve that. Because the thing is, if you're going to start with a very specific biofluid, one methodology is not enough. And if you have components in the end that affect the small RNA counts, then we have a problem, because I don't know what is associated with the vesicles and what is not associated with vesicles. But if your sample, for example, if your source doesn't have a lot of components other than the vesicles, it depends, because again, to maximize small RNA amounts, probably you're associating something that also has a high yield, right? A high yield meaning a high efficiency. So do you want to have a high efficiency, but then you have a low specificity associated to that, and ergo your RNA profile is clouded by other components that you have there? Or do you want to have something that has not as much small RNA, but at least you know what is there is associated with your vesicles? A bit dependent on what you want to do in the end.
Thank you. The next question is from Similori Babawale. he or she asks, apologies. A question about size exclusion chromatography. How does it compare in terms of purity if you separate your EVs in different fractions? And how pure EV sec is compared to different steps, and if there's an additional step post-sec that you can use to enrich certain EVs.
Okay, so in terms of size exclusion chromatography, this is, from my experience, size exclusion chromatography really shines in less complex biofluids. So I think if you were to put size exclusion chromatography in the, again, but this is the worst example that I could give, right? Blood plasma, not the best, because you have a lot of things that overlap in terms of size. However, actually, in my hands, size exclusion chromatography works pretty well with methodologies such as, with the biofluids, I'm sorry, such as cell culture supernatant. Because the complexity of the biofluid, so the components that are present there, are not so high that in the end you're going to have such a big amount that you're going to have like a very high amount of contamination from your sample. So actually, it was if that is your, again, my question is, what would be your source, right? So this really goes from the cascade. What is your objective? What is your source material? And then I can start thinking about some proper methodologies to go about in that. However, if you don't have the information based on what is the source material, or even what is the objective in the end, then it makes it very hard that you actually apply the tool that you want based on that.
Next question is from Yvette. She asks, could you point out the best technique to identify the origin of these EVs? I think that's a very tricky question. An idea was to use proteins specific to a cell type, for example, neuronal versus astrocytic proteins.
Yeah, so I guess we go again about it, but that it's basically, I think I like to put things into perspective in the way that we break it down into pieces. So you essentially, the point was that the origin of the vesicles. In order to determine the origin of the vesicles, you would need to know if they have specific compartments or specific components that you would expect from specific origins of the vesicles, whether they being exosomes, biogenesis, etc. Thing is that there's no specific markers right now. That's a bit of a problem. However, let's go about it this way. If you were to, in a specific scenario, in a cell scenario, completely inhibit the release of exosomes, and you only will have the exosomes or the microvesicles, and that's the only thing you have in your biofluid, then I would suggest, again, methodology that has a very high degree of specificity, because in the end, you will know that the proteins or the components that you're detecting are at least specific to the ectosomes that you're studying. This can be also vice versa. If you are to inhibit ectosome release and you have if you have exosomes only, the same thing can go about. So in specific scenarios, I think where most people want to do really biomarker analysis or want to do really something that is EV associated, I think rule of thumb, high specificity methods, so highly pure methods, because that really unlocks the claim that something is associated to vesicles. Whilst if you have vesicles and something else, you will never know if the effect that you're seeing is based on the vesicles or the combination of vesicles and something else, or just the fact that something else is present there.
Okay, there are some more questions from the same people. If you've got some more time, I can ask them. If not, we can stop. It's up to you.
It's okay. We can go. We can go.
Okay, so I'll go back to some of the questions we skipped at the beginning. So a follow-up question from Dalila Finelli. So she asks more about EV delivery and the mode used by EVs in communicating with recipient cells, and how best could we study these processes?
Yeah, okay, so the delivery of vesicles is, so I assume this is yeah, well, there's a lot of questions that I can then I have based on the question. Are the vesicles already there, and then you want to put something inside the vesicles, or the vesicles is something that is released from the vesicles or from the cell itself?
I think she means if there are EVs that you isolate from a cell and you're not really sure exactly what's inside them, if you were to take these EVs and add them to acceptor cells, what would be the best way to study this process?
Right. Well, so I think there's several publications based on for that as well that have tried to do some sort of delivery of it. So there's people that have done they've placed some tag on the vesicles, and then they check for the tag in the cell itself. There are people that put specific silencing RNAs in the vesicles, and then check if the RNA is silenced or if there's no or there's less RNA in the cell itself. There's multiple ways that you can go about this. Again, of course, in these scenarios where you want to study if the vesicle of the delivery is based on this, or how the impact goes, if you want to have only vesicles and actually say that the cell is affected because it's only the vesicles, again, high specificity methodologies. If that doesn't really impact and you want to see if actually the cells only take up vesicles, for example, despite the presence of something else there, a high efficiency method is maybe better as well. Because you have more vesicles, you have other components, but you don't care because that's really what you want to study is really, if you can, for example, visualize the delivery of this, then other components will not matter because you can just visualize the vesicles. So very study dependent as well, but achievable accordingly to the biofluid and your objective in the end.
Okay, and then I think this might be the last question from Marianne, asking about bioreactors and what you think about bioreactors to increase EV yield.
I am a fan of bioreactors most of the time. I myself I already thought about implementing some here in the lab as well. The problem about bioreactors that I usually have is how exhaustive it is for cells sometimes. So if cells are not, well, they either grow on top of each other. So if cells start to grow on top of each other and they start to expand more and more and more, then you have the problem that they get into stressful conditions. If they get into stressful conditions, you get the release of more components out to the medium. You have the release of apoptotic bodies because a lot of cells start to die because of the stressful environment. That is usually a problem on top of just the whole exhaustion of the cell itself. So usually that's my only caveat that I have regarding bioreactors. Everything else I am a fan of. So the construct of it, I am a fan.
Okay, thank you very much, Claudio, for your time. Thank you very much for the presentation. As mentioned to everyone who's still here, the talk has been recorded and there will be a YouTube video present on the SNEV YouTube page. Also, I think we will put the link to the paper. I think it was a Nature Reviews primer that he used. A lot of the information was present there, and that will also be present in the bio in the YouTube page. So once again, thank you very much, Claudio.
Thank you for having me.